Let's look at what we're doing this week in week 2. We're in module 2 and we're looking at the foundations of cell structure. We're looking at membranes and organization. We're trying to look at how the cell is organized, how it moves material within the cell. In your weekly objectives, it shows you that we're going to look at how the different organelles interact with one another. We're going to look at that endomembrane system. So within the cell, there's a whole bunch of different membranes that interact with one another and work to help materials flow throughout the cell and be transferred from one part to the other. We're also going to look at how foreign chemicals interact with cellular function. So if we look at something like a medication or a toxin, one of the reasons why they do have an effect is that they interfere with that cellular machinery. We're going to look at the membrane because the membrane is the gatekeeper to the cell. It allows certain things to come in, certain things to go out. That is also how cells communicate with one another and how different parts of the body communicate. But that's also one way that toxins and various other substances can get into the cell. So it's very important that we understand that membrane structure. We're also going to work on, from a more skill perspective, how to interpret graphs. In one of your assignments, you're going to have some graphs and you're going to have to describe what that graph is telling you. This is a very, very important skill that we have to develop as scientists because we often are looking at other scientists' results and papers, and that will typically be presented in graphical format, and we need to be able to understand what that is telling us. We'll also go over the differences between prokaryotic and eukaryotic cells. So first you have your learning materials. You'll have this weekly lecture. You'll be reading chapter 3 in the OpenStax textbook. You have some videos that go over membrane and transport. These really help visualize what is going on. It's much easier to see this in a video than it is in a static diagram. You also have some interactives. These will allow you to go inside of a cell and kind of see what's going on in a game type format. There's also some information here on antibiotics because one of your assignments this week will talk about how antibiotics work. This will help you with that assignment. And then there's also a science skill section. Remember, we're going to talk about learning to read graphs. This shows you a little bit about reading graphs and understanding them, and it also gives you a link to Unity's Com Hub, which gives you a little bit more information about citation, how to do research. Your first assignment for week 2 is a protein transport diagram. You're going to build a diagram showing how these internal cellular organelles work to build and then transport and then export proteins. So you're specifically looking at that particular function of a cell. You want to draw these components. You also want to show the steps of how these organelles contribute to this process. So it gives you a little bit of background. You're going to create this diagram either using Google Slide, PowerPoint. You can use this diagram. These need to be ones that you have developed. So you're not just taking a picture of a cell online and putting it in there. You are drawing it out because that's going to help you learn it better than just copying somebody else's photo. You also may do it by hand, but you need to make sure that it's legible when you upload it. You're going to illustrate this eukaryotic cell showing how these different organelles work together for this creation, transport, and export of proteins. You're going to use arrows to show the sequence of events, and you're going to label those. Next to your label, you're going to have a short description of what is happening in those different organelles. You want to make sure that your diagram includes all of these elements and how they relate to protein synthesis and transport. You're also going to write a 50 to 100 word paragraph describing your diagram, and in that you're going to state how and why you think that diagram demonstrates the efficiency of a cell. So talk about efficiency. What is it about that structure that you just drew that makes that eukaryotic cell efficient? And then you're going to predict what would happen if you went to one of those components within your cell and removed it. What if that component wasn't there? What would happen within that cell? That is your initial post. In your reply post, you will respond to one of the diagrams from one of your peers. So try to find somebody who has not been reviewed yet. You're going to give 2 specific comments for the diagram that they posted. One should highlight a specific aspect that you appreciated. The other should be a possible suggestion for refinement or improvement. Then you're going to use an AI tool of your choice to find a toxin that hinders protein synthesis or export from cells. And you're going to research how that toxin interacts with the cell to prevent that process. You're going to download your peer's diagram. You're going to use a drawing tool on their diagram to indicate in a different color of ink how that toxin would interact with the cell. And then you're going to repost your diagram, which is a modification of their diagram, with a description of how that toxin impacts the cell activity. And you should integrate any sources that you use. Make sure that you include both an in-text citation and an end citation. Every time we have a citation, we have to show in our essay or our paragraph or whatever where we use specific information from that source, and then we also need to give a complete citation at the end. There are videos and a citation guide in the announcements for how to do that. Your next assignment for the week is going to look at osmosis and how environmental conditions can affect organisms by affecting osmosis. this. You're going to create a fact sheet that highlights how road salt influences amphibians, specifically thinking about how that salty environment would influence osmosis and their cell structures. You're going to be talking about osmosis, semipermeable membranes, the different biological processes by which we move things across membranes. You're going to think about how a salty environment would influence that and create a fact sheet. In your fact sheet, you need to talk about the process of osmosis, describe how water moves through that semipermeable membrane. You want to use in-text citations where you use sources. You're going to create an image of a frog, and you're going to have an arrow indicating how water will move either into or out of the frog if we place that frog in a hypertonic solution, which is one that has a higher salt concentration than the inside of the frog's body. You're going to write a statement that explains how the frog could maintain homeostasis in both a high and a low salt concentration. So if I were to put it into a highly salty environment, what would happen inside that frog's body to keep it alive? And the same thing if I were to put it in, say, distilled water with no concentration of solutes. How would that frog manage to stay alive in that situation? Last thing you're doing here is looking at these graphs from this article. You're going to write a figure caption that explains what is happening in these graphs. You need to very specifically say what is happening. Now, one of the things it also tells you is you don't have to worry about these little A's and B's here. The little A's and B's are essentially telling you the statistical significance of each one of those points. We're not going to go into that yet. You don't need to worry about the statistics behind it, but you do need to describe what each of these graphs is showing you. Underneath, you have your grading rubric. Always make sure you look at both the instructions and the rubric. Your next assignment is a presentation on antibiotics to a high school audience. You're going to focus on the different types of cells and their evolutionary history. You're going to look at potential drug targets that are found in both prokaryotic and eukaryotic cells. When we talk about drug targets, we're essentially saying, where does that drug go? What does it interact with? So is it changing receptors on the cell membrane? Is it getting inside the cell and influencing something with the mitochondria? What is it actually doing? You also want to think about and highlight the development of drug resistance. Many pathogens become resistant to our medications. We obviously have to then develop new medications because the old medications are no longer effective. When we look at this evolution of drug resistance, that is a huge issue that we have to face, and it is a problem that we will continually have to try to overcome. Down here, it gives you a little bit further instructions on this presentation, what sections you should have. So you'll have 5 sections. You'll have 1 to 2 slides per section. Look at each of these where you are describing prokaryotic and eukaryotic cells. You're describing the endomembrane system, how they work, why that increases efficiency, compare and contrast plant and animal cells. Then you're looking at potential drug targets found in both prokaryotic and eukaryotic cells. What do we mean by drug target? What are 2 potential drug targets found in both prokaryotic and eukaryotic cells? Then we want to think a little bit about unique structures in, say, bacterial prokaryotic cells compared to eukaryotic cells, because this is how we can attack pathogens. If the bacteria has something that our cells don't, then if we have a drug that can target what the bacteria cell has that ours don't, it can attack the bacteria without attacking our own cells. So that's always an issue with drugs, right? We have to somehow destroy the pathogen without destroying the host. And so that is one of the things it's looking for here. What types of structures does a prokaryotic bacterial cell have that our cells don't, so that we can target those? And it's asking you to justify with specific scientific evidence. So one of the things you want to start doing, instead of just making very general claims, is you want to back it up with data. That is what science is about. We don't just say, hey, trust me, this is going to happen. We show you data that support that statement. In section 4, you're going to talk about what happens when someone takes an antibiotic. You're going to follow that antibiotic essentially through the organism. And then you'll have a conclusion, which will reveal why antibiotics are effective to target bacteria. Throughout this, you will want to make sure that you use in-text citations and the APA format end citation. So you'll have a slide at the end that lists your citations, and on your individual slides where you use information from those, you will indicate that in a parenthetical reference. Again, you have your grading rubric down below. After you make your slides for your presentation, you will record yourself presenting it. Please make sure that you present it in a way that you would find appealing. So don't just read off of the slide. And what you want to do here is to use Kaltura to record your presentation, unless you have some other mechanism for doing that. There's a link here that will take you to the use of Kaltura. It's really quite easy. It shows you some instructions here. If you have any questions during the week, please let me know. I hope you have a great week.